Just over a week ago, an FDA advisory committee voted 8-6 to open a legal supply line for BPC-157. The thing they voted on it was for treating ulcerative colitis. Almost nobody buying BPC-157 is buying it for ulcerative colitis, though. The whole human evidence base the FDA could find for that use is just a single trial, and it was reported as a conference abstract and not a peer-reviewed journal. In it, the drug was given as an enema, nobody takes it like that, at 80 milligrams once a day for 2 weeks. But tendonitis is closer to why most of you have probably heard of this stuff, and tendonitis was on the nomination list too. But the FDA didn't evaluate it for tendonitis, and the reason sits in one line in a footnote. The nomination didn't include enough evidence to assess it, and the agency couldn't find a single clinical study of BPC-157 in those patients. So the committee opened a door using evidence for a condition people don't want it for, and the condition they do want it for never got looked at. I'm Dr. Jordan Feigenbaum, and this is the Barbell Medicine Podcast. I've posted about peptides a few times this year, and each time the posts blow up, particularly the comment section. Now, people make real arguments, and a few of them have come up over and over again, so I'm going to try to work through those and see if we can find some common ground. But before we do any of that, two things about what I'm claiming, because I think both tend to get misread, and I want to nail down my position. The first one, I am not telling you that BPC-157 does nothing. I don't know that, and neither does anybody selling it to you. Nobody's done the study that would answer the question anyway. The FDA's own reviewers put it carefully, and I'm using their words, not mine. There is a lack of evidence to make a conclusion on effectiveness. Not it failed, they're just saying that we can't conclude. There's no evidence. Those are different senses and different claims, and I'll keep saying the second one. We just don't have the evidence. Also, a note on how that first sentence tends to land. When I say a thing hasn't been tested, a lot of people hear me saying that they were stupid to try it, but that's not what I'm saying at all. There's no evidence is a statement about what's going on in the research world. It's not a statement about your judgment. You didn't decide not to run the trial. Somebody else decided that, and there's money talking behind that. The second thing that I'm saying, I am not telling you what to put in your body. Several folks said, look, it's not my business. And on the personal question, that's fine. But that's not what happened last week. What happened is that a federal advisory committee voted to build a legal supply chain for compounds that have never been tested for the things people are trying to use them for. That's a public decision made by named people, and I think that's fair game. Let's start with the objection that isn't about peptides at all, because I think it's underneath all of the other claims. Somebody commented that after what we watched the medical professionals say during COVID, that they have zero trust in the entire system. Look, I'm not going to pretend that those years went well. Guidance changed, sometimes quickly, and plenty of it got delivered with more confidence than the evidence behind it could carry. That part's on us. Updating an answer when the data changes is the thing that's supposed to happen with science, but from the outside, it looks identical to having been wrong the whole time. Almost nobody stopped to explain the difference while it was going on. Where I get off the train is the conclusion. Guidance changing is not evidence that somebody was hiding something. But if what you took away from all of that is that you should always ask who has money in a particular answer, follow the money, well, look, I think that's the right instinct, and this vote is a great place to apply it. In September of 2023, the FDA moved close to 20 peptides into what's called Category 2, which means they have significant safety risk or there's a concern for it. That shut down compounding of them essentially overnight. Peptide medications like BPC-157 and TB-500 disappeared from licensed supply chains within weeks, and demand went straight to the gray market. Then in February of this year, the Secretary of Health and Human Services went on a podcast and called the 2023 restrictions illegal. His argument, in his words, was that the agency isn't supposed to restrict these without a safety signal, and he says that there isn't one, and that the FDA isn't allowed to look at efficacy and use that to move something into Category 2. He also said, and I'll quote this part because it's honest, "We created the gray market." Now, to be clear, what the FDA weighs here isn't just safety. Category 2 isn't a separate program with its own rulebook. It's a holding spot. Somebody nominates a substance for the compounding list, and while the FDA works through it, they sort it. Enough information and no safety concern, it goes to Category 1 and pharmacies can keep using it. Enough information, but there's a safety problem, it goes to Category 2, and they can't. Not enough information to evaluate it at all, it goes to Category 3. Now, these peptides went to Category 2 in 2023, and the FDA's own language is pending further evaluation because the underlying question never changed. Does this belong on the list? That list is governed by an actual regulation, Federal rules have labels the same way that statutes do, and this one's label is 21 CFR 216.23. Title 21 is Food and Drugs, Part 216 is Pharmacy Compounding, and Section 23 is this list. It's a rule with the force of law, it's public, and you can pull it up on your phone right now. It names 4 things the agency weighs. 1. What the substance physically and chemically is. 2. Safety issues raised by using it in compounded drugs. 3, and this is the one that matters here, the available evidence of the effectiveness or lack of effectiveness. And then 4, whether it's been compounded historically. Effectiveness is one of the 4, so saying the agency can't consider it is saying they should ignore a quarter of the rule. Then in April of this year, 12 peptides came out of Category 2. Now the FDA doesn't go hunting for substances to put on the compounding list. Somebody has to ask. Anybody can ask, and you file the request with whatever supporting information you have. In practice, the people asking are almost always the people who want to sell it or use it. Compounding pharmacies, industry groups, that's just how the list gets built. And a pending request is what gives the FDA something to look at. That's what put these drugs in Category 2 in the first place. The request to look at these peptides went in all the way back in 2015. A compounding pharmacy network and a peptide industry group requested them. Then, in April of this year, both of them pulled their requests. With nothing on the table to look at, there's nothing left to sort, and the Category 2 label comes off usually within 7 days. So the restriction didn't come off because anybody looked at new evidence. It came off because the people who asked the question stopped asking it. That didn't make these drugs legal to compound. They didn't move to an approved list. They stopped being labeled a safety risk and landed in no category at all. And that's where they sit right now. That didn't make these drugs legal to compound. They didn't move to an approved list. They simply stopped being labeled a safety risk and landed in no category at all. And then the FDA held the hearing anyway. It's in the briefing document in a footnote under every single substance. This nomination was withdrawn by the nominator. However, the FDA is electing to proceed anyway. So nobody was formally asking for these to be reviewed anymore, and the agency took all 7 to the committee on its own. So let's look at who was in the room and compare it to who used to be. This committee has looked at peptides before. In 2022, and in the meetings around it, panels like this one reviewed peptide requests and turned them down. The same kinds of substances, same existing evidence, and the same answer. So the June 2022 meeting is the last look that this committee ever took before the personnel turned over. The 2022 panel was built out of academic pharmacy and drug safety. There was a pharmaceutical sciences professor from UConn. The acting chair was a former executive vice president of the Institute for Safe Medication Practices, which is an organization whose entire job is medication error. And there was a scientist from the U.S. Pharmacopoeia, the body that writes drug quality standards. The rest came from Duke, Hopkins, Harvard, Indiana. That panel and the ones like it looked at peptide nominations and voted them down repeatedly. But the roster from last week looks nothing like that. Now, there are 11 standing voting members. Standing means that they sit for a term and vote on everything that comes before the committee. 7 of the 11 are affiliated with wellness, longevity, peptide, or men's health businesses. So 7 of the 11 standing seats held by people in the business. And the evidence didn't change between those 2 panels. The FDA's own scientists reviewed all 7 of these substances this time and recommended against every one. The same professional staff position, same thin evidence base. What changed was who was sitting in the chairs. Now, before the meeting, the FDA also added 8 temporary voting members. 5 of them are actual subject matter experts, and each of those 5 is locked to a single topic. The liver specialist could vote on BPC-157 and nothing else. The neurologist could vote on CMAX and nothing else. And the dermatologist was limited to KPV and TB-500. Bringing in temporary members with expertise in the specific substance under review and limiting their votes to just that substance, that's standard advisory committee practice. It's not a trick, and Peter Lurie, who used to be an FDA associate commissioner, publicly called those temporary additions more qualified and less conflicted to what was there before. So on any given peptide, the specialist who actually knows that compound is 1 vote out of 15. 11 of those 15 are the standing members, and they vote on every substance, whether it's in their field or not. And 7 of those 11 make money in this space. The other 3 are temporary members who aren't tied to a single topic, the pharmacokinetics professor, the consumer rep, and the patient rep. So the specialist shows up for 1 vote and then goes home, but the standing members are back in that room for the next peptide and the one after that. So how did a panel of academics turn into a panel of clinic owners? It's nothing dramatic. This committee runs on a clock. Members serve fixed terms, and the old panel's terms ran out on schedule. Nobody got fired, and there's even a holdover. The U.S. Pharmacopoeia scientist from the 2022 panel, he's still the acting chair right now. The seats come open the way they always do. The question is, who got put in them? The reporting on the conflicts is from the Associated Press, and it found that across the roughly 20-member panel, at least 7 members had financial ties to businesses that sell or administer peptides. Nothing about the molecules changed between the panel that rejected these and the panel that passed them. It's the same compounds, the same evidence, and the FDA's own scientists reviewed all 7 and said no to all 7. The variable that changed was who was sitting in the chairs. Now, there are 2 ways to read this. One is that money bought the vote. People with a financial stake got seats and voted in their own interests. I can't prove that. The other is simpler. Whoever filled these seats picked people who already believed peptides work. Those people then voted the way they already believed. Nobody had to be bought because nobody had to change their mind. I think the second one is more likely, and it's the one that should worry you more, because it can't be fixed by anyone recusing themselves. Either way, the outcome is the same. A panel was assembled that would produce this result, and it produced it. So if you don't trust a system where the people making the call have skin in the game, good, let's use that skepticism here. Before I take the next one, let's talk about what actually got voted on. Let's start with compounding. Most drugs you take are made in a factory in a fixed dose in a form that the FDA approved. Compounding is the other path. A pharmacist takes raw ingredients and mixes a drug to order for a specific patient, usually because the commercial version doesn't fit. A kid who can't swallow a pill and needs it as a liquid, somebody allergic to a dye in the standard tablet, a dose that nobody manufactures. These are all legitimate causes going on for about as long as pharmacies have existed, and it fills a real need. The catch is that compounded drugs are not FDA approved, nobody reviews them, nobody runs a trial on them. The pharmacy is licensed by the state, and what protects you is that the ingredient going into the mix is a known quantity. So Section 503A of the Food, Drug, and Cosmetic Act sets 3 doors that an ingredient can come in through, and a pharmacist only needs one of them. Door number 1. The ingredient has a U.S. Pharmacopoeia monograph, which is a published official standard describing exactly what the substance is. Door number 2, the ingredient is already a component of an FDA-approved drug, so somebody has already vetted it. Door number 3, the ingredient is on a list the FDA maintains specifically for substances that don't have either of the first 2. That list is called the 503A Bulk Drug Substances List, and people shorten it to the bulks list. Now, BPC-157 has no monograph, and BPC-157 is not a component of an approved drug in any country. The FDA confirmed both of those in the briefing documents. So doors 1 and 2 are shut, and the bulks list, door number 3, is the only way in, which is why last week's meeting happened at all. The bulks list is basically an approved ingredient list. It says that a pharmacist can legally buy the raw ingredient and make something out of it. It says nothing about whether what they make does anything or is safe, because those were never the questions being asked. So that's the whole vote. 7 substances, 7 votes on whether each one goes on the ingredient list. So when you see FDA panel votes yes on peptides, what actually happened is a committee recommended adding them to that list. It's a recommendation, not an approval, and it doesn't bind anybody. The FDA decides, and then there's a public rulemaking process. By the pharmacy trade's own policy, that's going to take place somewhere in 2027 before anything changes. So nothing is legal today that wasn't legal the week before. Now, one more piece of the mechanics, and this is what makes the ulcerative colitis vote matter more than it looks. Once an ingredient is on the shelf, a clinician can write for whatever they judge to be reasonable. Off-label prescribing is broadly at a physician's discretion. So a colitis vote produces a tendon supply line in this case. Nobody has to exploit a loophole for that to happen because that's just how prescribing works. One claim surrounding this vote is that the bulks list exists specifically for compounds that haven't been through the rigorous testing a commercial product goes through, but that haven't been shown to cause unwarranted harm either. So, the claim goes, when I keep saying that we don't have studies showing that these medications work, I'm applying a standard that was never the standard. That's not quite right. The bar for the bulks list is genuinely lower than FDA approval. Nobody has to run a phase 3 clinical trial to get on the bulks list, and the FDA doesn't apply a threshold on any one of the 4 criteria we mentioned before. The agency explicitly balances the criteria against one another, substance by substance. So the claim is right that there doesn't have to be proven efficacy in order to get on the bulks list, but where it comes apart is the word never. Effectiveness isn't a bar that you have to clear, but it is one of the 4 things that the agency weighs. It's written right into their own regulations. So it's not that the standard doesn't exist, it's that it's one input among four instead of just the gate. And BPC-157 didn't fall short of that one alone. The FDA's reviewers found it insufficient on characterization, on safety, and on effectiveness, 3 of the 4. That's why the agency's own staff recommended no. And there's a sentence in that same regulation that almost nobody knows exists. That same regulation that creates the bulks list itself carries its own warning label in paragraph section D. It says that based on evidence currently available, there are inadequate data to demonstrate the safety or efficacy of any drug compounded using any substance on the list. So the rule that authorizes the ingredient tells you, in the rule, that being authorized is not evidence that the drug works. And the next sentence goes even further. If anyone tells you a compounded drug made from a substance on this list is FDA approved or endorsed by the FDA, that representation makes the drug misbranded under federal law. It's written right into the regulation. The people who wrote this anticipated exactly the marketing that's coming. So that's the regulation. Now, the argument I see more than any other is that nobody's ever going to test this stuff because there's no money in it. The version most people say is some flavor of, look, yeah, I'd like if it was fully tested, but there's no financial incentive for Big Pharma to do that. It's a natural fragment, you can't patent it, so the trial's never going to happen, which means demanding a trial is demanding something that can't exist. So those are 2 claims stacked on top of each other. Let's start with the patent one because it's the piece people repeat most, and it's mostly wrong. To start, the premise that these peptides are natural and so you can't patent them is wrong. None of this is natural in any sense that matters. Every vial of BPC-157 in circulation is synthetic, it's made in a factory. And nobody's extracting anything from anybody's stomach, and we don't actually know it comes from a stomach or from humans at all. It's a manufactured chemical, same as any other manufactured chemical. And the patent claim doesn't hold either. You can patent a manufacturing process, you can patent a formulation, a delivery system, a specific salt or ester, you can modify the molecule, which is exactly what somebody did with C-Max, bolting a tail onto a fragment of ACTH so it doesn't break down as fast. Heck, Merck patented MK-677 in 1995, and it wasn't invented out of thin air either. And patents aren't the only way that a company gets paid. There's regulatory exclusivity, a period after approval where the FDA won't approve a competitor's version regardless of patent status. There's orphan drug status, which adds 7 years for rare diseases. There are entire business models built on running trials on old off-patent molecules and getting exclusivity for the new indication. So the you can't patent it argument is doing a lot of work, and it really can't carry the weight. Now, if one of these reliably fixed tendons or low back pain, there's going to be a way to make money on it, and somebody in Big Pharma probably would have found it. So that's the patent part, but I think there's another piece in this argument that's actually better. Almost everything in your medicine cabinet was studied to treat a disease or lower the risk of one. That's how the approval system is built. A company picks an indication and runs trials in people who have it. The trial exists because the disease exists. But these compounds are a bit different. They're mostly sold to healthy people who want to recover faster, feel sharper, age slower, or otherwise be better. Making a healthy person a little better is usually not an indication. There's no disease to enroll and no endpoint that the FDA recognizes. So for a lot of these compounds, the trial never happens. And it's not because anyone's hiding anything. There's just no version of the study the approval system knows how to run. And notice what this means. These are drugs, they're medications if you prefer. Untested ones sold outside of the system that generates evidence, which is a different thing from being supplements. That's a real structural problem, especially if you're looking to treat healthy people. But it isn't what happened with BPC-157, because a pharmaceutical company did develop it and they did try to test it. A Croatian company called Plevna ran it under a few different designations over the years: PL10, PLD116, PL14736. I promise you I'm not making these up. It's just letters and numbers. There's a phase 1 clinical trial in healthy volunteers looking at safety, tolerability, and how the drug moves through the body. And in 2005, a multi-center randomized double-blind placebo-controlled phase 2 trial. Now, both of those come with the same caveat. Neither was ever published as a full paper in a journal. Both exist as conference abstracts. So when a safety group says there's no published phase 1 data on this compound, they're right. And it's the same thing I mean when I say there's no published randomized controlled trial in humans. The trials were run, or at least one was. The results were never written up in a form that anybody could check. That on its own is actually kind of damning. The entire human record for a compound that maybe millions of people are injecting is 2 meeting abstracts from a company that quit. But whatever shape the write-up took, a pharmaceutical company ran the trials. Randomized, double-blind, placebo-controlled in humans. That's the study design people in my comments tell me will never happen, and it happened 21 years ago, and then the company walked away from it. So let's be clear about what that settles, because there are two versions of the no money in it argument. The first is, nobody would ever spend money studying this compound in humans. Well, that one's dead. Pleva spent years and multiple trials on it, and subsequent companies who have acquired this drug have tried to do the same. The second is about the specific use, and that's a better version to me. Fine, somebody studied it for the gut disease because there's a market there, but nobody's going to study it for a sore tendon or joint pain, except that's not true either. Musculoskeletal pain is one of the biggest drug markets there is. Companies have spent decades and enormous amounts of money on tendon and joint pain. NSAIDs, cortisone, platelet-rich plasma, hyaluronic acid injections, full phase 3 programs on an antibody for osteoarthritis pain. Some worked, some didn't, some got pulled, but the money is there and the trials get run. So the question was never whether anyone would study a compound for joint or tendon pain. They do it constantly. The question is why nobody has run one on this one. And as of this year, somebody's trying to. There's a phase 2 trial registered and recruiting in acute hamstring strain. That's the study that would answer most of what every lifter listening to this actually wants to know, and it's happening. Now, if this pattern feels familiar, it should, because I did a post on MK-677 that generated the same argument in my comments. Big Pharma won't patent it. They don't want you healthy. It's being suppressed. Now, nobody in my comments actually thought MK-677 was an awesome drug, but the story goes, Merck designed and patented the drug in 1995. They ran it through multiple large randomized trials over 15 years in Alzheimer's, in hip fracture recovery, in postmenopausal osteoporosis. And in 2011, a data monitoring committee stopped a trial early because congestive heart failure was running at about 6.5% in the drug arm against about 2% in the placebo. Merck shelved the adult program. And then somebody else picked it up. A company called Loomis took the compound along with Merck's original data and is running it in children with growth hormone deficiency. There's a placebo-controlled phase 3 trial going on right now. So that's not suppression. That's a compound that failed in one population and is being properly tested in another one, where the disease is real and the trade-off is different, which is how this is all supposed to work. So both compounds people tell me could never be tested are ones a pharmaceutical company already tested. Some of these get studied, and then the company walks away from what it finds. Now that doesn't cover all 7, because some of them are new enough that nobody's had time to study them. But the "there's no money in it" argument doesn't explain either group. There's enormous money in musculoskeletal pain, in sleep, in the beauty industry. And if one of these worked, somebody wants to own it. I don't have a clean answer for why nobody's running these trials, and I'm not going to invent one. But what I do know is that a missing trial is a missing trial. However it went missing, it doesn't tell you that the drug works. And there's a reason it might stay missing, which is the part of this vote that actually costs us something. Peter Lurie made the argument at the hearing, and I think he's mostly right. Any company that might fund a real efficacy trial now has a reason not to, because compounders can sell the compound without the study. So ask what happens to that hamstring trial's enrollment and funding when the drug becomes legally available for musculoskeletal injuries without anyone finishing the study. The tragedy of this vote is that it makes a study that could settle all of this much less likely to get run. Now, one of the biggest areas of pushback I see is that people are saying that they've used this stuff and it worked. The claim goes that thousands or even millions of people have gotten out of pain with BPC-157 or otherwise seen benefit with a different peptide on this list, and so we can't possibly ignore all of that and be honest at the same time. That's a good point, so let me be careful here with what I'm saying. When it comes to BPC-157, I'm not telling you that your injury healed on its own and that you imagine the rest. I'm telling you that the "I took it and got better" and "it works" are two different claims, and the gap between them is one of the oldest problems in medicine. And that gap is widest in pain and injury for a specific reason. Tendon and joint pain, they flare up and they settle down on their own. And people go looking for a fix when pain is usually at its worst, which is exactly the moment it has the most room to improve no matter what happens next. And on top of that, most people who start a peptide or any other treatment for pain change other things at the same time. How they're exercising, how they're sleeping, what they stop doing or start doing, and their expectations, because you just committed to something serious that you believe might work. So the improvement is real, but what caused it is genuinely unknown. And this isn't just a hunch or my own pet theory, because medicine has run this experiment on itself using fake surgery. In 2002, the New England Journal of Medicine published a trial where patients with arthritic knee pain were randomized to one of 3 groups. Two groups got real arthroscopic surgery, which was the standard operation at the time, and hundreds of thousands of those were being done each year. The third group got a sham surgery or a fake surgery. Patients were sedated, and the surgeon made real incisions in the skin. Then the surgeon splashed saline around so it sounded right and sewed them back up without doing anything inside the joint. They followed all three groups for 2 years. The fake surgery patients improved just as much as the real ones. They had the same pain relief, and they had the same ability to walk and climb stairs as the group who got the real operation, all the way out to 2 years. The people who got the incisions in their skin and a theatrical, dramatic performance reported that their knees felt better. Now, I'm not saying that they were lying. Their pain genuinely dropped. It just had nothing to do with what the scalpel did inside the joint because the scalpel didn't do anything. And it wasn't just a fluke. You can go all the way back to 1959. There was a common operation being done for chest pain from heart disease, where a surgeon would tie off an artery, the internal mammary artery in the chest wall, to supposedly reroute the blood to the heart. Patients swore by it. In 2 small trials, randomized people to either the real operation or a sham, where the surgeon opened the chest and closed it without tying anything off. Both groups improved about the same. Now, I'm not comparing BPC-157 to surgery. I'm just making a point. If a fake knee operation can produce 2 years of measured pain relief, then your elbow feeling better after an injection tells you very little about whether the compound in that injection did anything. The improvement is real, it's just that the attribution is unreliable. It's kind of like the rooster and the sunrise. Is the rooster causing the sunrise? And that's why we run placebo-controlled trials, because human beings cannot tell the difference from the inside, including me, including every surgeon who was doing those knee operations in good faith. Now back to the post I made on this vote the other day. A clinician commented about his experience in a patient with ulcerative colitis. This person was on dual biologic therapy and steroids and was headed for having part of his colon removed. He started BPC-157. A month later, he left the hospital off both biologics and off of the steroids, much to the surprise and likely celebration of his physicians. I can believe that happened. And if I'd have been in the room, I would have also been celebrating. But what makes it harder to interpret is the rest of the chart. That patient was on active treatment with biologics, and biologics have a delayed response window typically. Getting off steroids is the endpoint biologic therapy is aiming at, and ulcerative colitis remits and relapses on its own, which is why the disease is so hard to read from just a single case. And of every condition somebody could have written to me about, this was the one where the controlled test exists. Ulcerative colitis is the one indication where a real randomized placebo-controlled trial in humans of BPC-157 has been run. So we don't have to argue about it from anecdotes, and we can go look at the trial itself. The human trial was performed by Ruenzi and colleagues in 2005. It was a multi-center, randomized, double-blind, placebo-controlled phase 2 clinical trial of PL14736, which is BPC-157 under Pleva's old designation. 53 patients, a control arm, blinding, the whole kit and caboodle. It's the study people keep telling me will never exist, and they ran it 21 years ago. Now, I've said on this show before that BPC-157 has no published human randomized controlled trial data, which is true. We'll get to that. But suspend the apparent contradiction for just a moment, because the obvious read of a randomized trial is that we finally have an answer. We don't, and the reasons are worth going through one at a time. First, this study exists only as a meeting abstract in the journal Gastroenterology. A conference abstract is a poster and a paragraph. It was never peer-reviewed the way a full paper is, and the complete study was never published. So 21 years later, the methods, the endpoint definition, the inclusion criteria, and the statistical approach are still not available for anyone to check. The FDA says exactly that in its own document. But here are the numbers, straight from the abstract as the FDA had received them. 53 subjects with mild to moderate ulcerative colitis randomized 1-to-1 to either BPC-157 enema or to a placebo. 80 milligrams once daily for 2 weeks, and 46 of those patients completed the study. 3 withdrawals in the treatment arm, 2 in the placebo, which the authors attribute mainly to the colitis itself progressing. Now, the primary endpoint was a change in a disease activity index, described as a composite of clinical, laboratory, endoscopic, and pathology findings. The abstract never defines exactly what all that is. So one concept before the numbers, because without it, the numbers don't really mean anything. When a study reports a result, it also reports a range around it. That range is a set of values that fit the data they actually collected. And if that range includes 0, then no effect at all is one of the answers that their data is consistent with. So the treated group's disease score dropped about 3 points, and their range stayed entirely below 0. so those patients got better, and nobody's disputing that. The placebo group dropped about 1.5 points, and the difference between the two groups, which is the number that actually tests the drug, was about 1.5 points, with a range from about -5 to +1.5. Now that range includes 0, so the comparison that actually tests the drug against placebo didn't clearly show a benefit. And I can't even tell you whether 1.5 points on that scale would matter to a patient because nobody's published what that scale was. The authors called it a composite of clinical, laboratory, endoscopic, and pathohistological findings, and the FDA says that it was not clearly defined in the abstract. 21 years later, nobody outside the company knows how the primary endpoint was even scored. For context, the endpoint that ulcerative colitis trials typically use now is clinical remission on the modified Mayo score, and for a drug you'd take chronically, the FDA recommends at least a year of controlled treatment. This was 2 weeks on an index nobody can look up. This is where the proponents land, so let's name the move before it gets made. The change with the treatment group, on its own, cleared 0. People in the treatment arm got better. That's the number that gets quoted. But the within-group improvement is not evidence of a drug effect because ulcerative colitis relapses and remits on its own. People got better on the placebo, and people get better on nothing. That's the whole reason you run a control arm. And against the control arm, this missed. Now, to be clear, this isn't a slam dunk that BPC doesn't do anything. 53 people is a small study, 2 weeks is short, the study was exploratory, and an abstract is not a fair test of anyone's science. And the FDA's conclusion is careful, word for word. There is a lack of evidence to make a conclusion on effectiveness. That's different from saying it failed. So what does this trial establish about the vial in your gym bag? Nothing. Nothing at all. It's the wrong condition, given by the wrong route, in the wrong dosage. Start with the forms they wanted to sell, these peptide companies. Oral capsules, subcutaneous injection, nasal spray, transdermal cream, rectal suppositories. Well, the FDA went looking for human studies of BPC-157 given by mouth, under the skin, up the nose, or through the skin in colitis patients. They didn't find anything at all. The closest match is the suppository, and it still doesn't match. This trial used a liquid enema, and those don't reach the same places. A suppository stays in the rectum, but an enema can reach much further up the colon. So the single human trial doesn't line up with any product that was nominated, including the one it's closest to. Now, the rest of the human record isn't much better for BPC-157. 17 people with knee pain from a retrospective chart review at one clinic, and also some of those people got a second different peptide at the same time. 12 women with interstitial cystitis, single-arm study. 2 healthy people given an IV infusion. No control group in any of these studies. And here's the thing you notice reading the FDA's reference list. All 3 of those are first authored by the same clinician, published in the same alternative medicine journal. So outside of the 2 Plevna abstracts, the entire human record for this compound is one practitioner's uncontrolled case series. This podcast is brought to you by ButcherBox. One thing I try to stay consistent with is keeping enough protein in the house so I don't end up ordering takeout a few times a week. That sounds simple, but between work and everything else, the grocery store trip is usually the first thing that gets cut. And ButcherBox fixed that for me. They deliver grass-fed beef, organic chicken, wild-caught seafood, and more, all sourced to the standards they are transparent about. Every box is customizable, so you pick the cuts and the proteins that fit how you actually cook and what you're actually going to eat. It's like having a butcher on call who already did the sourcing homework for you. And let me just tell you, the quality is legit. I've been grilling a lot lately. It's summer here in San Diego, and the meat has been excellent. It tastes great. It shows up right to my door, exactly what I want, and I don't have to think about it. I don't have to make a trip. That's the whole point. It's an easy button for your food. Now, whether you're grilling on the weekend, meal prepping for the week, or just trying to get a real dinner on the table on a Tuesday, you've already got something good in the freezer ready to go. Go to butcherbox.com/barbell to get $20 off your first box, plus your choice of free ground beef for life, or free chicken thighs or top sirloins in every box for a year, with free shipping always. That's butcherbox.com/barbell, BARBELL. Be sure to use our link so they know that we sent you. This podcast is brought to you by Factor. The last few months have been a real time crunch for me. I've been finishing the book Signal, and between that, training, and everything else I've been doing, cooking has been a real challenge. And Factor has been the thing keeping my nutrition from going by the wayside. Having meals ready to go that I can heat up in 2 minutes and actually feel good about eating has made a real difference on the days where I just don't have those 30 minutes to stand in the kitchen, especially when it's late at night. Factor meals are chef-crafted and dietitian designed. They're ready to eat with no prep and no cleanup. They have over 100 menu items rotating every week, including new options like salmon burgers, shredded pork, and collard greens, plus add-ons like protein shakes and pumpkin cheesecake. Over 175 banned ingredients, so the stuff that you probably don't want in your food isn't going to be there either. I use Factor, and I bet you could benefit from it too, especially if your schedule gets tight. Food is usually the first thing that slips. This keeps that from happening. Head over to factormeals.com/BBM50off and use code BBM50off to get 50% off and one free breakfast item per box for a year while supplies last until October 31st, 2026. That's code BBM50off at factormeals.com. Again, BBM50off at factormeals.com. See the website for more details. This podcast is brought to you by Quince. End of summer is when I start thinking about what I actually want in my closet for the next few months. You know, not a full overhaul, just swapping out the stuff that's worn out and filling in some gaps. And that's what keeps bringing me back to Quince. They do everyday staples really well. Their organic cotton tees are soft and hold up wash after wash. Their cashmere sweaters are legitimately good for the price point, and they transition straight into fall. If you need pants, their premium denim and tailored chinos start at just $60, and the fit and fabric are on par with brands charging 3 or 4 times that. Ask me how I know. Now, everything at Quince is priced 50 to 80% less than similar brands because they work directly with the factories and skip the middlemen. You are paying for the materials and the construction, not a logo or marketing campaign. Now, one of my go-tos this summer has been their organic cotton mesh stitched sweater polos. I have it almost every color, and it's one of those pieces where you look put together, but you're actually comfortable, which is why these are going to stay in my rotation for the fall. And Quince goes way beyond just clothing. They've got premium bedding, bath essentials, cookware, travel gear. It's one of those brands where you buy one thing and end up recommending it to everyone. Both Lee and I have their sheets, and they're great. So upgrade your everyday. Download the Quince app for app-exclusive offers or go to quince.com/BBM. Get free shipping on your order and 365-day returns. Now available in Canada and the UK too. That's QUINCE.com/BBM. This podcast is brought to you by Figs. If you're in healthcare or you're in training for it, back to school season means you're about to spend a lot of time in scrubs. And Figs just dropped new colors and back to school styles. And right now, everything on their site is 20% off. Now, during my medical training, I found out that the standard hospital scrubs were not designed for anyone squatting, period. Bending over to examine a patient was like a trust fall, just hoping that the seam would hold. That's how I ended up trying Figs. They're cut with actual stretch, they fit like real clothes, and they survive both the job and the squat program. They're also antimicrobial, and they hold up to the laundry abuse that this job requires. They also have footwear, compression socks, and outerwear. And 20% off site-wide is the best deal they run. So if you've been meaning to replace your scrubs or stock up for a new rotation, this is the time. Go to wearfigs.com, get 20% off everything through August 11th. No codes needed. That's wearfigs.com. Hey, it's Jordan here. This is an ad for a subscription that removes ads, including this one, which is a good way that you never have to hear this again. And you can do that by signing up for Barbell Medicine Plus, which is now 25% off annually through August 13th with the code PLUSANNUAL. Now, the ad-free listening feed means this exact minute of your life, you'll never have to do this again. You also get member-only AMAs with Austin and I, plus bonus episodes, early access to our content, articles, and more. Plus, you get discounts on training templates, 10% off, 15% off consults, 25% off courses and seminars, plus member-only sales. If you're going to buy any of that stuff anyway, you now have solved the ad problem by shopping, which is not how I thought this was going to play out, but mostly it's how people support the show, and that support is the reason it keeps going. So sign up for Barbell Medicine Plus, use code PLUSANNUAL through August 13th. This has been an ad about itself. Now back to the show. So that's the evidence, and it's not great. The other argument I get whenever I call these drugs is that they're not, because BPC-157 is a fragment of a protein that your stomach makes, so it's natural. And a natural molecule isn't really a drug. Well, there are many drugs that are natural. Insulin is natural. Testosterone is natural. Thyroid hormone is natural. All three are drugs. Whether a molecule occurs in the human body has nothing to do with whether it's a drug and nothing to do with whether it's safe. Botulinum toxin occurs in nature too, and a few grams of it will kill you. And a small molecule made of ordinary parts can also be extremely powerful. Levothyroxine, which millions of people take every day, is a modified amino acid with iodine bolted onto it. It's simple, it's tiny, and it's boring on paper, but if you get the wrong dose, it can put you into atrial fibrillation. Size and simplicity just don't tell you anything about potency. And for BPC-157 specifically, the premise is worse because nobody's actually established it. BPC stands for body protection compound, and the claim goes that it's a fragment of a protein in human gastric juice in your stomach, but that parent protein has never been isolated or characterized in a human. The original description was a crude extraction in a patient, not a sequenced, reproducible protein that anyone can point to. And the 15 amino acid sequence in the vial isn't known to occur anywhere in nature either. Nobody has even identified the receptor for it. Nobody knows what it binds to or what it does once it's inside of you. And while I'm here, the word peptide is doing more work in this conversation than it really can support. A peptide is a short chain of amino acids, and in 2020, the FDA drew a legal line at 40. 40 or fewer, it's a peptide. 41 or more, it's a protein, regulated as a biologic. That number is a regulatory convention that somebody picked. Watch what it does. Semaglutide, what you know as Ozempic and Wegovy, is 31 amino acids long, so it's a peptide. Insulin is 51 amino acids long, so insulin is a protein by that rule, even though every clinician I know calls insulin a peptide hormone and has done so since training. Thymosin beta-4, the parent of TB-500, is 43 amino acids long, so that's a protein too, and MK-677 shows up in the same online stores as everything else here. And that one's a small molecule that Merck patented in 1995 with no amino acid chain in it anywhere. All of this is to say that none of this is new science. BPC-157 was first described in the early 90s. So why is it becoming a thing now? I think the answer is GLP-1s. Semaglutide and tirzepatide did two things at once. They made injecting yourself into a normal weekly habit. And on top of that, they worked visibly on people's coworkers and relatives. So a reasonable question formed in a lot of people's minds. If that peptide works that well, what else is in the drawer? There's a second thing that changed, and it's about how you get it rather than what it is. If you wanted anabolic steroids 20 years ago, you had to know somebody. There was a guy, there was a conversation you had to have in person with that person, and that friction did a lot because it made you decide out loud that you were somebody who was going to do this. You knew that you were buying a drug. Nobody was confused about the category. But none of that exists here. You order from a site with a chat widget and a discount code, and it arrives in a padded envelope in about 2 days. And that difference isn't because one of these is better studied. Testosterone is an approved drug with decades of trials behind it and a controlled substance schedule. BPC-157 has no approval anywhere in the world and 2 conference abstracts. The one with the evidence is the one that's hard to get. So what went away is the moment where you had to admit to yourself what you were doing. And the vocabulary finishes the job. Peptide sounds like something in a protein shake. Research chemical sounds like a lab, not a pharmacy. Short chain of amino acids sounds like nutrition. None of these phrases sound like the word drug, which is why I repeatedly call these things drugs. So let's just define it, because the definition isn't mine and it isn't a judgment call. Federal law says a drug is anything intended to treat, cure, mitigate, or prevent disease, or to affect the structure or any function of the body. That's it. That's the whole test. Read the second half again. Affect the structure or any function of the body. If you're injecting something to make a tendon heal faster, you're using it to affect the structure and function of your body, which means you're using a drug. It doesn't matter what it's made of, where it came from, or what the label says. Now, the FDA's own document calls these bulk drug substances and active pharmaceutical ingredients throughout, not supplements, drugs. Which brings me to the strongest argument I've heard yet, and it's the one the yes votes leaned on the hardest. People are buying this stuff anyway, from vendors that nobody regulates, so let them get it from a pharmacy instead. Half of that position, I think, is reasonable. The members who voted yes argued that patients are already buying this from unregulated vendors and that a compounding pharmacy testing for sterility, potency, and purity beats a guy shipping vials from overseas. The chief medical officer of one telehealth company testified that a colleague sent him a photo of a bodega in Queens selling peptides. The sourcing problem is real. I've written about it. I've talked about it on this very podcast. When researchers chemically analyzed 44 products sold as research chemicals through these channels, only about 4 in 10 contained what the label said. Some had no active compound at all. Some contained a different drug that wasn't on the label anywhere. And the FDA's own document backs up the danger of the current channel. The certificates of analysis in the nomination packages mostly reported purity and little else. No identified impurity profile, no microbial testing, no endotoxin testing. That's for a product proposed as an injection, and no endotoxin testing in an injectable is a real risk for sepsis. And there's a naming problem underneath this too. BPC-157 is a common name, not a United States adopted name. That's the formal system that exists so the pharmacist hands you what your doctor actually ordered. Aspirin has one, ibuprofen has one, BPC-157 doesn't, which means the name doesn't pin down what's in the vial. And there are at least 2 different substances sold as BPC-157. There's a freebase and an acetate salt. Different chemical structures, different registry numbers, and the FDA treats them as 2 separate drugs with potentially different safety and efficacy profiles. but both are sold under the same 3 characters and a number. This isn't a hypothetical. Both companies that nominated this to the FDA filed paperwork so inconsistent that the agency couldn't tell which one they were asking for. Their application said BPC-157. Their certificates of analysis were for the acetate. And then the FDA ended up evaluating both because it couldn't determine what had even been nominated. If the people whose business this is can't specify which molecule they mean on a federal filing, your pharmacist probably can't either. So yes, the current channel is dangerous, and I'm not going to argue with anybody about that. But I'm not so sure that compounding solves it. And here's why. Big compounding operations, the 503B facilities that make drugs in batches, report to the FDA twice a year what they've made. From 2017 through mid-2025, none of them reported a single dose of BPC-157. But that doesn't mean nobody was making it. Smaller pharmacies don't report the same way, and the FDA's own search turned up compounding pharmacy websites selling BPC-157, plus a concierge service selling injection kits for home use. 2 of the 3 adverse event reports in the FDA's file name a specific compounding pharmacy. So the picture isn't that this was absent from pharmacies, it's that whatever was happening wasn't reported and wasn't legal under the statute, because BPC-157 doesn't meet any of the 3 conditions that let a pharmacy compound with it. The case for putting it on the bulks list is that people are buying it from unregulated vendors, so let's move them to pharmacies. But some pharmacies were already selling it outside the rules. So people are buying it anyway includes people buying it from pharmacies that weren't supposed to be selling it in the first place. Which means the choice isn't between a gray market and a regulated one. There is no regulated one. There's a gray market that runs through websites and a gray market that runs through some pharmacies. And neither one of them is operating under a rule that says this substance is okay to compound. Listing it doesn't move anybody from unregulated to regulated. It makes one half of the gray market legal and it leaves the other half exactly where it is. And that's the part that doesn't fit the harm reduction label. Harm reduction means meeting demand that already exists with something safer. This makes the same product legal in the same places it was already being sold, at scale, with advertising. Safer would mean knowing what the drug does, which unfortunately isn't on the table. And if you want to know what this vote is actually about, watch the stock. The day of the BPC-157 vote, Hims and Hers jumped more than 10% intraday before closing up about 3%. The next day, the panel turned down one of the 7 peptides and the stock fell. Leerink Partners, an investment bank, called the vote a positive catalyst for Hims and estimated that the peptide business could be worth about $2.2 billion a year across telehealth, with Hims taking roughly a fifth of it. That's about $440 million a year for a company selling compounds nobody has shown to work. And Leerink isn't cheerleading. They're neutral on the stock, and their price target is below where it's trading. So even the analysts who don't think you should buy the company think there's real money in this. And the part that tells you this wasn't a surprise to anybody. Hims bought a California peptide manufacturing facility in early 2025, 18 months before this very vote. You don't reduce harm by opening a bigger store next to the sketchy one. Harm reduction works when the number of people using something is fixed and you're just changing where they get it. Clean needles don't create new injection drug users, but this is a wellness product marketed to healthy people who feel fine by a company that advertises on television. The number of people using it is not fixed. It's the thing that's going to be grown. So you're not moving a set group of people to a safer source. You're taking a substance a small number of committed people were seeking out and putting it in front of everyone who watches TV. And there's one move that would actually reduce harm. Run the trial so we know whether the thing works and at what dose. That gets less likely, though, the moment you can sell it without that trial being done. The people who voted yes leaned on the idea that a real pharmacy and a real prescription put a responsible doctor in the loop. But picture what that doctor is writing for. An unstudied injectable for use it was never tested on at a dose nobody has established. Finally, there's one last major objection to everything I've said so far. It sounds like I'm demanding trials for their own sake. It's more paperwork and more delay while people wait for something that might actually help them. So let me tell you what a missing trial actually costs by telling you 3 stories of how this has gone before. Let's start with fen-phen, because the structure is identical to what's in front of us right now. Fen-phen was a weight loss drug. 2 appetite suppressants taken together, fenfluramine and phentermine. Now, both had been approved individually and for a long time. Phentermine in 1959, fenfluramine in 1973, and both were cleared for short-term use only, a few weeks at a time, as a supplement to diet and exercise. The combination was never approved. It was never even submitted. No study demonstrating the safety or effectiveness of the 2 together was ever sent to the FDA, and the agency never signed off on using them long-term. There was nothing to rush through because nobody asked. What we had instead was one paper, a 1992 study out of the University of Rochester with 121 patients from a single center. They were followed for 4 years and fewer than half of them finished. About 40% were still in it at the end, and many of the ones who did finish regained the weight later. That study is the entire basis for what happened next. Doctors started writing for it off-label, which they're allowed to do, and it worked. People lost weight. By 1996, prescriptions for these drugs ran past 18 million. And it isn't that nobody raised a hand. Reports linking fenfluramine to pulmonary hypertension had been showing up in the literature through the 80s and 90s. There just wasn't good long-term data on either drug, and there were none at all on the two drugs together. Then, in 1997, a cardiologist at the Mayo Clinic published 24 cases. 24 women on fen-phen, no history of cardiac disease with damaged heart valves. The CDC eventually confirmed 113 cases. 111 of them were women. Median age was 44. Median time on the drug was only 9 months. Better than a quarter of them needed a valve replacement. The drug came off the market that September. So 3 things about that story matter here. The first is that valve damage is silent. You can't really feel it, and every one of those women felt fine until they weren't. There was no symptom to notice and no test that anybody was running. The thing hurting them was invisible from the inside, which is exactly what I mean when I can't tell you what to watch out for when it comes to these peptides. The second is who was wrong. 18 million prescriptions were written by physicians, real doctors with real training who believed it worked because their patients came back lighter and happier. Nobody in that story was stupid. Not the patients, not the doctors. Everybody was operating on exactly the kind of evidence people are operating on now, which is that it seems to be working and nobody's reporting problems. And then there's a third thing, which is the part that should sound familiar. While fen-phen was booming off-label, a company was trying to get a related drug approved, dexfenfluramine, sold as Redux. Same family, submitted as its own product. In 1995, an FDA advisory committee voted 5-3 against recommending it over concerns about pulmonary hypertension and possible neurotoxicity. So the FDA convened a second advisory committee. That one voted 6-5 in favor, approved in April of 1996. Nobody was quiet about it beforehand either. Neuroscientists had written the agency twice about neurotoxicity in animal studies. France had already restricted the drug because of the pulmonary hypertension cases. All of it got buried under the fact that this was the first new obesity drug in 20 years and the country wanted it. 2.5 million prescriptions went out by June of 1996, off the market by September of 1997, 16 months. A panel said no, a second panel said yes by 1 vote. The drug went out and it hurt people. The second one I want to talk about is Vioxx. Merck got Vioxx approved in 1999 and over 80 million people took it. The FDA reviewer who evaluated the application had already noted more heart attacks and strokes on the drug than on placebo. In 2000, the Vigor trial found 5 times the heart attack risk against naproxen. Merck's explanation was that naproxen protects the heart, not that Vioxx damages it. When the trial was published in the New England Journal that November, 3 heart attacks in the Vioxx group weren't in that paper. The FDA had the full data on its own website, though. In February 2001, an FDA advisory committee looked at it and said there was clear evidence of increased cardiovascular risk. They recommended that the label say so. The label was changed in April of 2002, and the reason for the delay is the part that should stop you. The FDA had to negotiate wording with Merck because the agency didn't have the authority to just require it. Their deputy director for new drugs later told the Senate it would have been helpful to have that power. Meanwhile, Merck put out a press release titled Merck Confirms Favorable Cardiovascular Safety Profile of Vioxx. The FDA wrote back calling the claim simply incomprehensible. It took until 2004 when a placebo-controlled trial was stopped early for the drug to come off. David Graham, an FDA scientist, testified to the Senate that Vioxx caused somewhere between 88 and 140,000 excess cases of serious coronary heart disease. He says he was pressured to soften his findings. Here's what Vioxx should actually teach you, and it isn't that the FDA can't be trusted. The FDA got it right. Their reviewer flagged the heart attacks before approval. Their advisory committee said there was clear evidence of harm in 2001. Their own scientists ran the study that quantified it. Every piece of the agency's scientific judgment was correct and roughly on time. But what they couldn't do was act on it because they had to negotiate the wording with the company selling the drug. Their deputy director had to go ask the Senate for the authority to require a warning. So the problem isn't that the FDA doesn't know anything. The problem is that the industry sits on both sides of the table and the agency's scientific judgment is the part that's optional, which is exactly what happened last week. FDA's career scientists reviewed all 7 of these substances and recommended against every single one. The panel voted yes to 6. The agency's own scientists lost again. in the agency's own building to a committee stocked with people who sell the product. So if you don't trust the FDA because you think that they're captured, you and I are looking at the same evidence. I just don't think the answer is buying an unstudied drug from a website. Okay, the third one, and it's the closest to home because it's about compounding specifically. In the fall of 2012, a compounding pharmacy in Massachusetts called the New England Compounding Center shipped contaminated steroid injections across 20 states. 64 people died. More than 750 were infected with fungal meningitis and spinal and joint infections, and for some of them, the treatment never ended. Now, that was a licensed compounding pharmacy, not a gray market vendor, not a guy shipping vials from overseas, a real pharmacy making an injectable, and it killed 64 people. And I want to be exact about what that does and doesn't say, because I'm not standing here telling you that compounding pharmacies are dangerous or kill people. Most of them are careful, and most of what they make is fine. What it says is that there are two separate ways this can go wrong, and the pharmacy only addresses one of them. The first is that the product isn't what it's supposed to be. It's contaminated, the wrong potency, wrong molecule. That's the NECC failure, and it's the one a good pharmacy is built to prevent, though NECC is the proof that they don't always. The second is that the product is made perfectly and nobody knows what it does. A clean vial of a compound with no monograph, no established dose, and no efficacy data is still a clean vial of an unknown drug. So get it from the pharmacy instead only answers the first problem. It says nothing about the second, and the second is what this whole episode is about. Congress responded to NECC in 2013 with the Drug Quality and Security Act, and that law built the compounding framework we have now. Federal oversight of large compounding pharmacies, the outsourcing facility category, the 503A structure everybody was arguing about in that hearing room last week exists in its current form because 64 people died. And every bit of it is about manufacturing, sterility, testing, oversight, reporting, necessary work, and it fixed a real problem. But none of it asks whether the drug actually works. That question wasn't what went wrong at NECC, so the law that came out of it doesn't address it. Which means when somebody tells you a pharmacy makes this safe, they're describing a system built to catch contamination. Contamination isn't the thing I'm looking for answers about for BPC-157 and these other peptide drugs. I want to know what they do. While it is true that there aren't good studies showing that these peptides work, people often turn that around and say that there aren't good studies showing that they're harmful either. To be clear, we don't have safety data is not the same claim as it's probably fine. So let's try to sort it out when it comes to BPC-157. First, the things we actually know. The animal work that was submitted includes a 28-day repeat dose study, and in it, the compound moved clotting times in both directions and pushed up liver enzymes, blood sugar, and triglycerides. On the human side, the FDA's adverse event database has a handful of BPC-157 reports. One is logged as life-threatening, and the reason listed, quoting the entry, is product sterility lacking. Another is a person who stacked BPC-157 with TB-500 and developed spreading darkening of the skin and gums, and the darkening came back when they dosed again. That return of symptoms on a secondary exposure is the closest thing in the whole file to the compound itself doing anything. Second, the things we know we don't know. We know that there's no long-term human safety data. We know that there's no carcinogenicity studies. The proposed mechanism people cite is angioneogenesis, growing new blood vessels after an injury, and that's a growth signal. The long-term studies that would tell you what a growth signal does over years were never run. Now, to be fair, BPC-157 did test negative for mutation in the standard genetic damage assays, and nobody has evidence that it causes cancer, but the study designed to look at this has never been done. And then there's a dose-response problem. In several of those animal studies, doses 1,000-fold apart produced about the same size effect. 10 nanograms per kilogram and 10 micrograms per kilogram each produced comparable results. The FDA's phrasing is that a dose-response relationship could not be established and that the molecular targets have not been identified. In simple terms, if the response is the same across a 1,000-fold dose range, it's hard to know whether you're watching the drug or just looking at noise. And I should say, a large share of that preclinical literature traces back to one research group in Croatia. Read the agency's reference list yourself, and you'll see the same senior author over and over and over again. Independent replication is thin to non-existent. But that doesn't make the work wrong. It just means that one lab's results haven't been checked by another lab. Third, the unknown unknowns. When physicians put a patient on a real drug, we can usually tell them what to monitor. For example, I can name the blood test and the symptom for the medications I'd consider using. For BPC-157, I can't, because nobody has established what shows up when something goes wrong over the years. Nobody has even identified the receptor that it uses. You can't predict the side effects of a molecule when you can't even name its target, and you can't screen for a problem when you don't know what the problem would even look like. So when somebody says, look, nobody's getting hurt, for BPC-157 specifically, they're close to right, But I wonder how much of that is because you usually only find harm when somebody's looking. And for this compound, nobody is. There's no registry, there's no doctor filing a report when a gym buddy's shoulder injection goes sideways. The absence of case reports in a population nobody is following could easily be a measurement problem. And when you widen the search out to the whole class of peptide drugs, the picture changes. In April, ECRI and the Institute for Safe Medication Practices published a joint white paper on exactly these compounds. Their finding on BPC-157 is blunt. 35 of 36 published studies were done in animals, with no published phase 1 human safety data and no controlled human efficacy trials. Across the class, they document real signals. A phase 2 trial of CJC-1295 was halted after a participant died of a cardiac event, and no one has picked it up since. Melanotan-2 has case reports on melanoma, including mucosal melanoma. There are case reports of kidney dysfunction and rhabdomyolysis, and at least one death following intravenous administration. Then the sourcing data. Earlier I told you about 44 products where only 4 in 10 matched their label, and that was about labeling accuracy. This is a different measurement, and it's worse. Analytic testing of 10 gray market peptide products found purity ranging from 5% to 75%. Not 5% off, 5% pure, and arsenic and lead contamination running up to 10 times the international toxicity limit for an injectable drug. Now I'll note who's saying this because it connects back to something from earlier. The Institute for Safe Medication Practices is the organization whose former executive vice president chaired the 2022 version of the FDA's committee. The people who used to be in that room are now writing white papers from outside of it. Now, to be clear, the ECRI and the ISMP support the advisory committee process. They think it's the right mechanism. Their objection is to what happened in April, when the classification changed without any new evidence being presented. Everything I've said so far has been about BPC-157, because it's the one most of you are asking about. But 6 other substances went in front of that committee, and the reason to run through them is that the answer is the same every single time. TB-500, it's sold for muscle and tendon repair, and the one most often stacked with BPC-157. A 7 amino acid fragment of thymosin beta-4. It was nominated for wound healing, but the FDA found no adequate human effectiveness or safety evidence for that use. The same characterization problems as BPC-157 and the same immunogenicity concern for an injectable. It's also on the WADA banned list, but it passed 8-6. KPV, a 3 amino acid fragment, lysine, proline, valine, snipped off the tail of a hormone called alpha MSH. It sold for inflammation and gut conditions, and lately it's jumped into skincare. The FDA's finding was blunt. The human clinical evidence was absent. Not modest, absent. It passed 8-6. MOTC, a 16 amino acid peptide your mitochondria actually encode, it's marketed as an exercise mimetic, which is the exercise in a vial pitch. Human clinical evidence, again, absent. Also on the WADA banned list. And this is the one where the panel kind of wobbled, 7-5 with 2 abstentions. So the compound with a purely performance pitch and 0 human data is the one that made even this panel flinch. CMAX, a 7 amino acid fragment of ACTH with a small tail bolted onto it to slow its breakdown. It's marketed for focus and cognition and popular in the nootropics world. CMAX is a registered drug in Russia, sold as nasal drops, so its supporters argue that there's more data on it than others. But the indications nominated here were stroke recovery, migraine, and trigeminal neuralgia. The FDA found the data insufficient and flagged a bleeding risk from the peptide's anticoagulant properties. It passed 8-5 with 1 abstention. Apidolon, a 4 amino acid peptide originally built off a pineal gland extract, sold as anti-aging on the claim that it activates telomerase, the enzyme that maintains the caps on the ends of your chromosomes. But telomerase activation is not obviously a good thing. Switching it on is one of the things cancer cells do to become immortal, and there are no carcinogenicity studies. So the marketing hook and the safety concern are the same mechanism. It passed 7-4 with 1 abstention. DSIP is a 9 amino acid peptide first pulled out of rabbit blood in the 1970s, named for the deep sleep brain waves it seemed to bring on. It's pitched for sleep and for opioid withdrawal. This is the only one of the 7 that the panel turned down. 6 in favor, 7 against, 1 abstention. The FDA had said the evidence didn't support insomnia, narcolepsy, or opioid withdrawal as indications. And that's the tell I keep coming back to. 6 of these got a yes. The one that got a no is the one where being wrong looks like an overdose. Everywhere else, the panel voted yes. Different molecules, different pitches, but one identical FDA verdict underneath all of them. Not well characterized, not enough evidence of efficacy, not enough safety data. The panel read that verdict 7 times and said yes to 6 peptides anyway. So that's the whole record. Now, what would I actually tell you if you were sitting across from me? Because the 2 situations here aren't the same. If what you're looking at is a research chemical, a vial labeled not for human consumption from an online vendor, then my answer is easy and it's a no. That's the channel where most products were mislabeled. Some had no active compound and some were spiked with a drug that wasn't listed. You have no idea what's in it and you'd be injecting it anyway. The compounding pharmacy version is a harder conversation. If these get listed and you can get a sterility tested, potency verified product from a real pharmacy on a prescription, then whether to use it becomes a genuine individual decision. I'd want you making that decision with a clinician who isn't the one selling it to you, which in this corner of medicine is not always a given. I'm not making that decision for you. What makes the decision hard isn't me deflecting, though. For most drugs, you weigh the known risks against the known benefits. Here, both sides of the scale are mostly blank, or at least foggy. The benefit for your specific use case has never been shown. The risk side is worse than unknown because we don't even know what to look out for. So you're choosing something that's kind of blurry on both sides. So if you decide to use, you should at least know that's what you're doing. There's a version of this I think is worth discussing directly. The it's my body, my choice, and a doctor telling me no is a doctor restricting my choice. Call it self-determination. The part of that I'll concede outright is the clinical dilemma. A patient tells you they're taking this regardless. You can fire them from your practice, or you can keep seeing them and monitor what you can. Firing them accomplishes nothing except making sure nobody with training is following them, which is bad. So you keep seeing them and do your best. That's the whole job. Where the argument breaks is that self-determination in medicine has a name already, and it's called informed consent. The word doing the work is informed. You have a well-established right to refuse anything. But what you don't have is a right to a decision made under information nobody's generated yet. When the risk side is blank and the benefit side is blank, there isn't a choice being protected. There's a coin flip being dressed up as autonomy, and the courts have been clearer about this than people think. Abigail Alliance was named after Abigail Burroughs, a young woman with head and neck cancer whose oncologist thought an experimental drug might help her. She couldn't get it, and she died in 2001, unfortunately. Her father built an organization and sued the FDA, arguing that terminally ill patients with no remaining options have a constitutional right to buy drugs that have cleared basic safety testing. The other case goes back further. It was about Laetrile, an apricot pit extract that was sold as a cancer cure in the 1970s. Terminally ill patients argued that the FDA's safety and effectiveness rules shouldn't apply to them because what does safe and effective even mean when you're dying? And in 1979, in a case called United States v. Rutherford, the Supreme Court ruled against them unanimously. The requirements still apply. Right to try is real, but it's a statute from 2018, not a constitutional holding. It covers drugs that have cleared phase 1 and are under active investigation, and it requires the company to agree to provide it. Nobody can force a company to hand anything over. So the picture people have of the law is kind of backwards. Dying patients with nothing left have gone to federal court asking for access to drugs that were further along than these, and they lost. There's no case that gets a healthy person a compound its sponsor abandoned 21 years ago, though. So I'm not the obstacle to your actual informed consent. The missing trial is. Now, some of you aren't injecting anything. Some of you are swallowing BPC-157 capsules for your gut. It's the same evidence gap, and then some. An oral capsule has to survive the exact system designed to digest it. It probably does nothing since getting a peptide to work orally requires a lot of sophisticated engineering, all of which is usually proprietary and very costly. If you compete in a tested sport, this isn't a judgment call. BPC-157, TB-500, and MOTS-C are all on the WADA prohibited list, banned in and out of competition. Pretty easy decision not to use if you're following the rules. And if the thing you actually came for is a tendon that won't settle, there's an intervention with real human evidence behind it. It's loading the tendon. Progressive, heavy, slow resistance training works. That's the thing shown in randomized trials to cut pain and remodel the tissue across the Achilles, the patellar tendon, and the elbow. And this is the part that should end the peptide pitch for tendon pain. The benefit people claim for BPC-157 is more blood flow to the area. Loading already does that. Load a tendon and blood flow through it jumps 2 to 3 times. And on top of that, the tendon lays down and reorganizes new collagen, which no injection does. Meanwhile, the cortisone shot and the PRP injection that clinics sell for the same problem don't beat a saline placebo once you're doing the loading, and cortisone comes out worse at a year anyway. The catch is that loading is slow. Tendons adapt over weeks and months, not days, and there's no version of this that's fast. Which is a real cost, and I understand exactly why an injection sounds better. You're in pain now, the fix is months out, and somebody's offering you a vial that promises it to be done in weeks. If that worked, it would be the obvious choice, and I tell you to take it. It would also be worth billions. A shot that reliably heals tendons faster than loading does would be one of the most valuable drugs in orthopedics, and every company in the space knows it. That's the part that should give you pause. Nobody has shown it, and the incentive to show it has been sitting there the whole time. Meanwhile, loading does the thing the injection is supposed to do, and it does it right now. More blood flow to the tissue, plus new collagen laid down in the right direction. There's no shot in the world that does the second part. So that's what I tell you. Now let me tell you what would change my mind, because if I can't answer that question, I'm just the mirror image of the people selling this stuff. There's a phase 2 trial in acute hamstring strain recruiting right now. Say that trial, or one like it, gives the compound the way that people actually take it, at the dose they typically use, in the injury they actually have, against a real placebo. And say it shows a pretty good signal that it works and that it's safe. Then I change my answer, and I say so right here on this show. That's all it would take, a well-done study of the thing in the people who use it against placebo. It's the same study I've been asking about all episode, and it's never been done. So I think that the committee got this one wrong. 7 changes, and they got it wrong on 6 of them. Take BPC-157. What they reviewed was a condition almost nobody uses it for, on a single conference abstract that didn't beat placebo, for a substance the agency's own scientists say isn't well characterized. The tendon question, the one that sent everyone looking in the first place, was never evaluated at all because there were no human studies to evaluate. And that's the good one. That's the substance with the most human data on the docket of these 7. Read the FDA's verdict for the other 6, and it's the same sentence with the name swapped. Not well characterized. Not enough evidence of efficacy. Not enough safety data. 7 substances, 7 reviews, 1 conclusion, written by the agency's own scientists. The panel read it 7 times and voted yes to 6. Improving access isn't evidence, and this vote didn't generate any. The trial everyone tells me could never happen has already happened twice, and neither time did the answer come back affirmative. And the study that would actually settle this is recruiting right now. Last week made it less likely to finish. And this isn't over. There's a second meeting expected at the end of February in 2027 on 5 more compounds, including melanotan 2, which is the one with published melanoma case reports attached to it. Whatever standard got applied last week is the same standard those 5 are going to get judged against. Because if people are buying it anyway is enough to open a legal supply line, then tell me what that doesn't cover. It works for these peptides, it works for the next 5. It works for anything with a website and enough customers, which is how you know it isn't an argument. I'm Dr. Jordan Feigenbaum. Thanks for listening.